Welcome to another lecture by MedicoMedics, Microbiology and Infectious Diseases, Chapter 6, Parasitology, the study of parasites. So in this lecture, we will give you a general introduction to what are parasites, different types, its life cycles, the pathogenesis of parasitic infections, and we will mention transmission, diagnosis, treatment, preventing infections, some examples of parasitic diseases, and then move on towards a summary. Now, parasites are organisms that live in or on a host organism, obtaining nutrients at the host's expense. And we have different types of parasites. So we have Protozoa. These are single-celled parasites. For example, Plasmodium. And why is Plasmodium interesting? Because it causes malaria. We have Helminths. These are worm-like parasites. Examples, ascariasis, tapeworms. We have ectoparasites. These are external parasites like lice or ticks. Now, importance. Causes significant diseases in humans, animals, and plants. So, parasites, all in all, are these uninvited guests that live off their host, often making the host sick. Some, as we mentioned, live inside the body, like worms or protozoa, while others stay outside of the body, like lice or ticks. And why are these interesting? Because these are, for example, ticks spread Lyme disease. So all of this is quite relevant. Now, these rely on their hosts to survive, but usually they harm them in the process. Now, let's continue talking about these different types. So we said protozoa. These are microscopic, single-celled organisms, illustrated here. Examples, as we mentioned, Plasmodium leads to malaria. We have giardia, which causes diarrhea. Then we have another type, Helminths. These are multicellular worms, right? And the types could be roundworms or flatworms, like tapeworms, etc. Then we have these ectoparasites that live on the skin or outer surfaces. Examples, ticks, which spread Lyme disease, and fleas, causing itching and transmit infections. And what's interesting is that, indeed, parasites come in different forms, right? Tiny single cells, like Plasmodium, to larger worms like roundworms, or even these creepy crawlers like ticks. But each type of these has a unique way of surviving, so it causes problems for its host. Now, let's talk about the life cycles of parasites. So we have, first of all, stages, where we have the egg becoming a larva and then developing into an adult. This is a prime example of the pathway for Helminths. Then we have trophozoites. So the egg becomes a trophozoite, and then continues to become a cyst. This is a pathway for protozoa. Another important concept is hosts. These are differentiated into definitive host and intermediate host. So in the context of parasites, the term host refers to the organism in which the parasite lives and from which it derives its nutrients or protection. And these hosts are categorized based on their role in the parasite's life cycle. So when we talk about a definitive host, this host is the organism in which the parasite undergoes sexual reproduction. And this host is critical for the completion of the parasite's life cycle. So, for example, in schistosoma, humans serve as a definitive host because the adult worms live and reproduce sexually in the human bloodstream. All right? Now, what about intermediate host? The intermediate host is the organism in which the parasite undergoes asexual reproduction, right? So, no sexual reproduction, but rather asexual reproduction or other developmental stages, but it does not do it sexually. So, it is essential for the parasite's development and transmission to the definitive host. So, for example, again, for schistosoma, snails are the intermediate hosts where the parasite undergoes larval development. And so, understanding life cycles is indeed crucial for controlling infections, specifically due to the way these sustain and develop. Now let's turn our attention to the pathogenesis of parasitic infections. Some mechanisms of harm include the stealing of nutrients. So, for example, tapeworms absorbing food from our intestines, damaging tissues like Plasmodium destroying our red blood cells. Another is triggering immune responses, so severe inflammation from schistosoma eggs. And what could be some symptoms following it? Well, malnutrition, or anemia, fever, organ damage, etc. Now, what about the transmission of parasites? So some modes of transmission include ingestion, so through contaminated food or water like giardia or tapeworm eggs. We have vector-borne spread, so spread by insects like mosquitoes, leading to malaria. Skin penetration, so larvae entering through skin, like schistosoma. Direct contact, so close contact with infected individuals or surfaces, for example, lice. Now, let's make this somewhat boring lecture a bit more fun and clinically relevant. So let's try and connect different parts of the lecture thus far. So we talked about, for example, mosquitoes, right? And let's start with the mosquito and let's end with malaria, or the process of having malaria. So what happens is that this mosquito that is itself infected with something called Anopheles mosquito, or it's called that, bites a human being, okay? And it injects spirozoites into our bloodstream. Then these spirozoites or sporozoites enter our liver, and they infect our liver cells, also known as hepatocytes, okay? And what happens is that they then multiply to form something called merozoites. So from sporozoites to merozoites. And these merozoites are then released into our bloodstream. Now these merozoites infect our red blood cells. All right? And they multiply and cause these red blood cells, okay, to rupture. And this releases then more merozoites and toxins into our blood because they were multiplying, remember? Now, the rupturing, this is where it becomes clinically important because the rupturing of these RBCs occurs in several cycles and lead to the symptoms of fever, for example, chills, sweating. These are classical malaria symptoms. And this continuous RBC destruction leads to eventually severe anemia. So essentially, we have now a reduced oxygen carrying capacity of our blood. Why? Because oxygen transports through blood and binds to hemoglobin in our red blood cells. So, what are then the complications of all of this? Well, the accumulation of parasitized RBCs eventually blocks capillaries, causing even organ damage, leading to, for example, cerebral malaria. And eventually, severe anemia or organ failure or cerebral complications, all of these could lead to death. So what started as something very innocent, a tiny little mosquito, a mosquito bite, spread and multiplication, entering into our blood cells, multiplying and rupturing, leading to fever, leading to red blood cell loss, leading to anemia, leading to severe anemia, eventually even leading to death. So that's a big picture of the why parasites and the study of parasites, etc., is crucial. Now, this was just an overview. We go into the details of the malaria in our malaria lecture. But for now, hope you get a brief overview of why we're studying this, why it's important to know the mode of transmission in a parasite, to know it's vector-borne, that it's spread by insects like mosquitoes for malaria. Beautiful. Let's go on. Now, let's briefly mention some ways to diagnose parasitic infections. So methods include microscopy, so identifying eggs, larvae, or protozoa in stool, blood, or tissue samples. Serology tests, so detecting antibodies against parasites in our blood. Molecular methods like using PCR to detect parasite DNA. Some practical examples could be a blood smear, so identifying Plasmodium in malaria, or a stool test finding Giardia cysts or Helminth eggs. And then what is the treatment of parasitic infections? So we use drugs like antiprotozoal drugs. These treat protozoan infections like chloroquine for malaria. Anti-helminthic drugs like albendazole, these are used to treat worm infections. What are some challenges that we're confronted with? Well, drug resistance in parasites. Difficulty in treating parasites that hide inside our cells. So, all in all, treating parasites involves medicine that either kill the parasite or stop it from reproduction or reproducing. But indeed, some parasites do develop resistance or hide in places where medicines can't easily reach. Then some ways to prevent parasitic infections include, for example, hygiene, so hand washing and proper sanitation, safe food and water, cooking food thoroughly, avoiding contaminated water, vector control, so using insect repellents or bed nets for mosquito-borne diseases. Vaccination. However, it is limited in availability, but malaria vaccines are, for example, in development. Now, let's just repeat some examples of parasitic diseases and mention some new ones. So, under protozoan diseases, we have indeed malaria caused by Plasmodium, transmitted by mosquitoes. We have amoebiasis caused by Entamoeba histolytica, leading to diarrhea. Then we have Helminth diseases. So we have ascariasis. These are large intestinal worms. Schistosomiasis caused by blood flukes. We have ectoparasitic diseases like Lyme disease spread by ticks. Now let's just briefly touch upon the connection between fungi and parasitology. So some parasitic organisms have fungal-like characteristics, like Pneumocystis jirovecii in immunocompromised patients. So treatment and diagnosis often overlap with fungal infections. So, in summary, then, parasites are diverse and can cause significant harm to humans. Understanding their life cycles, mode of transmission, and treatment is crucial for prevention. Global efforts are making strides in reducing parasitic infections. And that's the end of chapter 6. Continue now to chapter 7.